This is so cool. This is a website that's devoted to the genomics of insects called Flybase. The particular chromosome we're looking at is from a fruit fly. This chromosome has 21 million bases. You can see them across this level. The website gives us the ability to look at whatever part of this 21 million bases we want, kind of in whatever detail we want. The 21 million bases is here, and our magnification is across this part. Where we are on the 21 million bases is determined by this little red line. So as we move this little red line up and down, we're moving up and down on the 21 million bases. And there's a zoom function similar to other software packages actually, which is reached through this drop down. The zoom gives us a focus of anywhere from 100 base pairs to 500 kilobases, which is 500,000 bases, which is 500,000. So we can look at it really with any magnification that we want. There's a page up, page down function, which is found on these arrows. So you can click over, click downstream and click upstream as you please and look at different parts of this chromosome. So let's say we actually want to look at something specific. We can come up to this website and download the actual bases that are associated with the window that we're looking at by simply hitting go. And if we do that, then it opens up this screen and gives us the actual bases that we're looking at. I've never done it for more than about 1,000 or so, but I guess you could do it for 500,000 or whatever, and it would give you an actual printout of the bases. Let's take a quick look at this part of the screen. Down here we see what the actual genes are. If we put the pointer on one of these sites, then we get a drop-down window appears which tells us what the gene is, tells us what they know about it, and gives us as much detail as there is. And there's both a lot of information, and you can see by looking at these windows that there's also a lot of uncertainty associated with it. We notice that some of them are pointing one direction and some of them are pointing the other direction. Genes which are pointing to the right are on the chromosome as we're looking at it. Genes that are pointing to the left are on the complement pointing the other direction. So it's really rather interesting. The genes can be in either direction depending upon how the promoter found them. When we go to download the gene, if we want it downloaded as a reverse complement, all we do is click this little button, and then when we download it, we'll get the other side. A lot of research in molecular biology starts with this kind of approach. There are many websites, quite a few websites that give us this type of information. The biggest one is the NCBI website, which is the National Center for Bioinformatics. But there are others. This one is the Flybase website.